Today I'll be talking about scaling microbial dynamics with Bayesian nonparametrics. It's sort of a fancy way of saying all the things that Martin was talking about. We'll be talking about Dirichlet processes and some of the modeling components for the main talk. Okay. So what we'll get out of this primer, what we'll talk about are information not found in Travis' main talk, which is some of the mathematical backgrounds in terms of the terminology concepts you'll need to know to understand the main talk. And there are some parts that are going to be a little fast or even glossed over, which is the algorithmic motivation and sometimes the intuition for why things works or why we did things a certain way. So the story starts out with this so-called Lotka-Volterra model, which is a popular choice and sort of go-to choice for any time that people are trying to learn interactions or trying to model microbial dynamics. Now, it's generalized in the sense that when you first learn about Lotka-Volterra, it's 2 dimensions and it's just 2 variables. But here we're going to consider n different individuals. So up there it should say dx sub I dt. So the scalar form you can write it down as the ith component's derivative is equal to x sub I times this linear combination of all of the x's. So here, x sub I, think of x without the subscript as a vector, and x sub I is supposed to be somehow the abundance of the ith organism in your system. And you can sort of rewrite this in vector form. So the idea here is that the big circle with a dot in the middle is component-wise vector multiplication. So you're essentially saying for the ith organism, you're going to scale it by this linear, or rather affine transformation of x, and that's going to be your rate of change at each point. So an example that's useful to work with for people who might have never seen this before is that you might want to think about this as an analog of logistic growth. So if you consider that differential equation where the rate of change for x is equal to this quadratic function, x times r plus ax, let's say you set the initial conditions at 0 to 1/2. Well, depending on what r and a are, you can get different kinds of rates of changes for your solution, where ultimately there's this asymptote at 0 and at some well-defined carrying capacity, so which is what the horizontal lines are for each color. And you can see that changing the values of R and A changes either the rate of growth or the overall carrying capacity, or both, depending on the ratio between R and A. And this one, the example is useful because it essentially resembles this vector form over here, and it might be a little harder to recognize in that form over there. So if you haven't caught it already, the matrix A here on the right-hand side, the diagonals are A sub i's, and the off-diagonal entries are the BIJs. And in literature, we often give names to these for the reasons that I was talking about earlier, which is the R sub i's are called growth rates, and we often, when modeling microbiomes, we assume that's positive. It's somehow an intrinsic component of the organisms or the colony of organisms where if you just sort of leave them be, they'll just grow over time at an exponential rate. And the AIs are usually things that we assume to be negative, and we call that the self-limiting coefficient. And the reason it's negative is that, well, we usually like to think about organisms that can grow to a certain capacity but can't grow beyond that. Somehow, like if there's a finite number of resources that's available in a system, and somehow it's not supposed to be able to grow past what's possible given the finite number of resources. And the important thing here for this talk, and even it's even the title of the slides, which is the off-diagonal terms BIJs, we call pairwise interactions. And so that essentially governs the effect of the abundance of organisms in the system that's not the one that's described on the left-hand side of the derivative. And when you first study Lotka-Volterra, when you might first work with a toy example on the left-hand side over there, where you have a classic predator-prey system, and this is somehow unnatural because you have the cyclic behavior, but that's not why it's unnatural. It's unnatural because you often have to assume that the diagonal terms are zero, diagonal coefficients of the matrix. And for a more typical example where the self-limiting coefficients are negative, you might have more behavior that looks more like the right-hand side, where you have some abundances in the initial conditions, and they sort of do this semi-oscillatory behavior, but essentially stabilize over time. And the right-hand side is something that's more common and sort of a system that you'll find out in the wild. And well, the question that we want to think about here is not the scenario where we're given coefficients and we're trying to simulate a system. We're going to try to think about the inverse problem, where we're given time series observations of a system, and we're going to assume it's locked to Volterra. And we're going to try to find parameters that give rise to the observations, or are highly likely to give rise to the observations. So here, the goal is we're going to think about it as estimating the parameters of GLV equations given time series observations that provide the best fit. And I put that in quotes because somehow that's loose and it's up to interpretation. And we'll think about what this is. Oh, there's a question that says you have to assume, sorry, the diagonal terms are zero in the predator-prey model. You mean if they aren't zero, it doesn't oscillate at all? So yeah, so it's not that they don't oscillate at all. It's, when you see classical systems, they'll oscillate, and they'll oscillate because they have zero diagonal coefficients. It's somehow a simpler version. So I am not 100% sure under what kinds of circumstances or what kinds of coefficient manifolds give rise to oscillations in higher dimensions. But it's not a necessary condition, no, especially if you're considering systems of 1,000 or, it's something that's bigger than 2 or 3, for example.
I would imagine even in the classical system that if you stayed, that you could introduce small quadratic terms that wouldn't really affect the dynamics. Sorry, small diagonal terms that wouldn't really affect the oscillatory dynamics. I could be wrong, but I would imagine that as long as the oscillation stayed away from the carrying capacity, that they wouldn't really have an effect. They would only have a second order effect, and that the main effect of the oscillations would stay.
Right, yeah. And based on literature, so I'm not going to talk about this in these slides, but essentially, one popular way of thinking about it is somehow there's a stable manifold somehow, where if you, in higher dimensions, it's more likely that if you start with at a point and you let the system run, it's going to stabilize to a certain manifold and not escape that manifold. And that sort of in and of itself is not oscillatory in n dimensions, but it might be oscillatory in some submanifolds, for example. So I mean, it's an interesting point, and it's actually very hard to analyze, and there's lots of literature about this. Okay, so again, for the inverse problem, we often have this system that we start with, and it's often helpful to linearize it in this sense. So we're going to divide by x on both sides. And we know that the derivative of 1 over x times dx dt is equivalent to the differential of the logs. So this is linearized in the sense that the right-hand side is linear in x. And what often happens, computationally speaking, is that on the left-hand side we'll discretize it in an Euler fashion. So we'll have 2 different time points, and we'll compare not the difference in the abundances, but the difference in the log abundances, and say that the right-hand, that's somehow supposed to be equivalent to 1 iteration of this dynamical system plus maybe some noise. And this is more or less a heuristic. And when you talk about this linearized system, you can apply standard techniques such as linear regression, maybe with lasso ridge, and we'll talk a little bit about this. And there's a question that says, should the left-hand side be a vector equation? So I wrote down the equation for the ith organism, but you can actually simultaneously write down for all the organisms. I assume that that's what you should be doing to obtain the full system. But we'll talk a little bit more about this later as well, which is there's many coefficients you have to learn. So it's sort of an implementation choice as to whether you want to stack it in vectors based on organized by organisms, or you want to solve the whole thing. I mean, so there's a slightly different take on this. If you want to get into the sort of nitty-gritty details, is you can alternatively start with the stochastic differential equation with multiplicative noise, where you have this differential equation that we started with, but again on the right-hand side you're going to add the abundance times some white noise term, and if you do this, then the discretization above is interpretable as an Euler step or an Euler-Maruyama integration step for SDEs. But either points of view sort of suggest that the same technique applies. And here's kind of where we sort of thinking about the practical questions. So the previous technique I just talked about is useful sort of in toy examples or very small systems where very precisely that there's maybe something on the order of tens to 100 organisms. Somehow when we think about the microbiome, there's not tens to hundreds, but maybe thousands. And in that scenario, this technique doesn't really work that well in the sense that it's very computationally burdensome. So if on the right-hand side, this picture is this famous hairball network by this paper in Nature by Raul Venkatesan et al. And the point is that, well, the parameters in a GLV system, well, per organism, there's n+1. There's the growth rate diagonal plus the interaction terms. And overall, there's n plus n squared total variables they need to estimate. So if you kind of do the math about like just the common crux of how many parameters there are, you actually need at least this many measurements in time series, at least n+1, right? And you don't often have that many. And if you were to somehow if you somehow have the resources to learn all the coefficients, well, you're stuck interpreting this kind of hairball network where you have to manually or computationally check somehow like all the different interactions that happen. And the precise answer to how long it takes is it's O of n cubed. It's the cost of matrix inversion per organism. And the point is that it doesn't scale to systems with lots of organisms. So there's this other question of how do we actually obtain the observations at each time point? And for that, I'm just going to postpone it and suggest that you attend Travis's main talk. But for this primer, we're essentially going to leave it off by saying we're not going to directly observe XIs. We're going to see noisy measurements of it. But for now, it's going to suffice to just assume that we observe directly the XIs, right? Ultimately, that's where we want to go. By the way, any questions so far? Okay. So the technique I mentioned is linear regression, and it should suggest something natural to try to reduce, at least from the interpretation side, of how to reduce the complexity of the model. And just to be clear, when I use the word complexity, I don't mean that the model is somehow simpler or easier. What I actually mean is bit complexity. So the model is less complex or more complex in the sense that it takes fewer bits to represent it on the computer. So the first thing that you would try is just try regularization, lasso or ridge. The problem with this is that it doesn't address the runtime scaling issues. It's still essentially a matrix inversion problem, no matter what optimization scheme you use. And you can also you can also ask which one's better. And there's a paper that essentially suggests that maybe for GLV systems, lasso is generally better than ridge, but it's sort of up in the air why. Now, we do want to address this runtime scaling, and so maybe here's a different idea: is that we're studying not a GLV system that exists in a vacuum. We're studying microbiomes, and so there ought to be organisms that maybe are phylogenetically similar or maybe serve similar functions in the gut, for example, if you're studying gut microbiomes. And so, the idea here as a tantalizing question is that what if there's a permutation of the rows and columns, or rather there's a way to group the organisms in your system so that the interaction matrix here could roughly look something like a block matrix, right? Like all the organisms in a group interact in the same way to all the other organisms in the system. Now, this is, if you can do this, this is good, because now you're not stuck with n squared parameters. Yeah, you're not stuck with n squared parameters. You're stuck with a much smaller system where you only need to learn as many interactions as there are the square of the number of clusters. Now, that's all well and good, except we don't know the blocks. So maybe in our regression, kind of like how we enforce Bayesian versions of lasso and ridge, we could enforce a prior on your regression such that somehow it encourages division into blocks, and you can explicitly model the GLV dynamics this way. And here, a priori, you don't actually know which organisms are meant to be clustered with which, and you don't even know how many clusters there are. Right? So these are important things to keep in mind. And here's an example of how one might, or how we chose to do low complexity and Lotka-Volterra system. And the idea is that, well, we're going to start with the original equation. And again, how the picture before suggested it, we're going to assume that the matrix is somehow composed of blocks, right? So if the coefficient's not going to depend just on the organism itself, but rather on the block that it's in. And for the purposes of both talks, we're going to call these interaction modules, or I might also use the word cluster. And the idea here again is that if a couple of individuals are in one module, then it's going to interact in the same way across all of the individuals in that module to every other bug. So this one arrow is the interaction for all three colors to all three of these. So okay. And here's the prior that we're actually going to place on these objects, which are Dirichlet processes. And well, we should talk a little bit about what Dirichlet processes are. Well, what is a Dirichlet process? I'm going to talk about what the parameterizations actually mean. It's not so important at this exact moment. And the answer is that it's a probability distribution over probability distributions. So it's probability distributions in particular over discrete or countably supported probability distributions. And the analogy that I always like to use when talking about Dirichlet processes is that it's a distribution over random histograms. And there is a formal definition to this. Think like Gaussian processes. And we're not going to talk about it. We're going to skip straight to the construction because I think they're more insightful. And there are essentially two known equivalent ways. There are a couple more, but these two are actually the most insightful. And the first construction is the Chinese restaurant process, is what it's called in the mathematical community. And the idea here is that, well, here's a restaurant, and there's a bunch of tables, and in fact, potentially infinitely many tables. And at each table are sitting, some known number of people already. And here, a new person comes in, and this new person is highlighted in red, comes in and tries to decide which table to sit at. Now, this person wants to be popular and wants to sit at a table with lots of people already in it, but he's going to do this probabilistically. So he's going to sample a table based on constant proportionality to how many people are already sitting at that table. So if there are 3 people at the first table, then you're going to apply a weight to 3 out of the total number of people you've seen so far. But you're going to add an additional weight, alpha, which is meant to be positive, to sitting at a new table. And the point here is that, well, how does this translate to random histograms? Well, think about where the tables are physically placed, for example, on the real line as a sort of a cartoon depiction. And the point here is that, well, what if the tables were distributed according to some background distribution H? And so that's what the parametrization alpha H there up top means. And now you're going to think about this as a random histogram in the sense that, at each of these randomly sampled points, theta 1, theta 2, so on, distributed according to H, you're going to assume a height that's equivalent to the proportion of people sitting at each table. And this is going to converge to the Dirichlet process in the sense that if you do this infinitely many times, you're going to get many different weightings. But with probability 1, you're going to get something countably supported. Now, the second construction is the stick breaking process. And this is something that you'll see in a lot of implementations of Dirichlet processes in inference. And the idea is that, well, we're going to start with a wooden stick of length 1. We're going to start chopping it off. So the way we're going to chop it off is that we're going to sample a bunch of numbers called beta. We're going to label these as beta i's. And they're going to be distributed according to a beta distribution with parameters 1 alpha. And remember, for beta, 1 is the weight you're going to give to heads, and the second parameter is going to be the weight you're going to give to tails, the probability of tails. And the idea is that, well, okay, we're going to sample beta 1, and we're going to chop off the length of the stick proportional to beta 1. We're going to do this again. So in the second step, what we're going to do is we chopped off this little piece. And from the remaining part, we're going to chop off the part that's proportional to beta 2. We're going to keep doing that, so on and so forth. And if you do this infinitely many times, and this, it's sort of the same idea here where, for each stick, and you've decided the length of it at that point, you're going to put it on random positions, and the positions are going to be determined according to this background distribution H. And well, how do people use Dirichlet processes in the wild, and why are they useful? And the answer to that is that, well, it's a non-parametric. It's a parametric way to represent non-parametric families of distributions. So again, we're thinking about Dirichlet processes as random histograms. And here we're going to think about, okay, sort of switching gears slightly temporarily for a little bit. Think about this data here that's empirical, and we're going to try to model it using a Gaussian mixture model and ask, well, if we want to fit a Gaussian mixture model, usually you have to specify the number of components. But how many number of components do we pick to even model this in the first place? Well, you can stare at it and say, okay, it looks like it's a good fit for 3, but there's also maybe an argument for 2. So you don't really want to be stuck deciding this. So here's an example of what you can do. You can assume that the observations are normal. It's a mixture of Gaussians, but the number of components is, well, okay, there's theoretically infinitely many components, but the weighting of the components is random, right? And maybe you can decide that, okay, I'm only going to look at the components with weight at least 10 to the minus 3 or something, right? And if you try to do this, well, it actually yields something very interesting, which is, you often yield 2 components, but sometimes you'll get posterior samples which include 3 samples or 3 components. And here it's actually 4, even though it's slightly hard to see. There's a question that says, what are the gammas? So I assume that you're referring to this. I don't see it anywhere. Maybe the gamma distribution. So yeah, so it's a probability distribution that's meant to well yeah, it's a probability distribution that's meant to serve as maybe a prior for the alpha itself, right? So the y gamma. So
yeah, so I think in this example, this is just a toy example we put together. the gammas for the parts in the normal distribution are probably just for conjugacy to make it simpler. You can, an inverse chi-squared is often used for the parameters in a normal distribution. And actually for alpha here, it's gamma, but that's actually not what we use in our model. It's like a mixture of, I'll actually reference it. It's Escobar and West, like 1995, for an efficient sampling scheme to have a prior on alpha so that you're not actually picking alpha, because alpha actually indirectly controls the number of clusters. And once again, this is a simple example. In our actual construction, at each time step, there actually is like a finite number of clusters, and alpha kind of controls the number of clusters indirectly, because alpha controls how likely you are to go into a new table versus a table that already has people. So here, yeah, the gammas for the lambdas and tals are probably just conjugacy. And for alpha, we actually use a slightly different one that's simple to sample from. But good question.
Yeah, and actually, Travis brings up a good point, which is, the choice of alpha, the gamma distribution here, allows for an algorithm that he was mentioning, which I'll show later. And it's actually makes it nice to sample the posterior from. So alpha given, let's say, the number of components, beta, that you see. Yeah, good question. But again, like this is not the model that we use necessarily for this whole thing. So in terms of how do we apply this for the GLV system? Well, here's sort of a toy version or a stripped down version of the model that Travis is going to talk about. And it's a generative model on discretized time points. So the construction here is that, well, we're going to start with this prior. We're going to use the stick breaking version. And this random distribution, we're going to treat it as the assignment, the probability distribution over assignments of clusters to each of the organisms in the system. And we're going to use the same system that I talked about earlier, where the interaction coefficients have this block structure. And maybe we can go even one step further, which is, the coefficients are meant to be random. They also have priors. Again, as I mentioned before, the self-limiting term and the intrinsic growth rates are meant to be positive. So you might want to put truncated normal priors on them. And the interaction terms and the noise terms are meant to be normal. And these normal distributions are all chosen for conjugacy reasons, essentially. It's going to make computing posterior distributions nice. And well, now that we've put this prior on it, maybe we should stop and think, how did this actually help? Well, here's a fact about Dirichlet processes, which is if you take n discrete samples according to a random probability distribution from a Dirichlet process, you can actually show that if you take your n samples and bucket them by value, the number of clusters unique clusters that you'll see is on the order of log n. So, to make this more concrete, if you have 1,000 organisms, if you have n organisms, you should expect to end up with log n clusters. And so the regression problem that you started with, which was size log n when you had to estimate n square parameters, now you're in a much better situation where you're estimating log n square parameters. And this is in theory, but let's say you implement this in practice, and you'll see that the actual expectation depends heavily on alpha. So think about what it means. What the alpha parameter is doing, think about what it means to increase alpha. So going back to the Chinese restaurant process, for example, if alpha is very, very large and the limit as alpha goes to infinity, each individual is going to pick their own table. So each person is going to be at their own cluster. Now, the other fact is that, well, as I mentioned, because of the priors that we picked, the conditional sampling is relatively easy. And in particular for Dirichlet processes, you can actually design a posterior sampling algorithm because there are some very simple equations that I'll show you that kind of like what you see with Dirichlet distribution as a prior for coin flips. And these algorithms are designed using both perspectives. So using the Chinese restaurant process and the stick breaking process. And it's the equations that I'll show are very reminiscent of this. Now, in general, the way the Chinese restaurant process helps is that, well, if you think about a scheme where you think the clusters are random, but in an MCMC setting, you might want to maybe pick one organism at random and resample what cluster it's in. Well, the probability for the transition for the Markov chain that you'll design is exactly according to the Chinese restaurant process for the first time. So you're going to pick each cluster according to the size of the existing cluster. But then because we're doing it in a Bayesian setting, we're going to think about, OK, we're going to reweight these size of the tables with the model likelihood of the observations. And in the GLV setting, the model likelihood would include things like the interaction coefficients and the self-limiting term and so on and so forth. Now, the other half is that, well, remember we put a gamma prior on alpha, and this is why we did this. Well, there's actually a very nice sampling algorithm for sampling the posterior. So the idea is that, well, you're going to sample alpha, but using an auxiliary variable eta. And you see this beta distribution, and it turns out that if you sort of do this auxiliary trick, you can resample alpha according to gamma, and you do this for many iterations. And this itself is, if you stare at it for a little while, it's an MCMC algorithm, right? For one single step, you're going to want to resample an alpha, but to resample an alpha for a single step, you're going to want to employ like a mini Markov chain to do that for one step. And this is due to Escobar and West in '95. It's a very interesting paper to read, and somehow it's all of these steps are very natural if you stop and write down what the distribution looks like. Okay, and let's try to implement this, right? So all these ingredients that I told you about is supposed to give you a very simple Markov chain. And so here we're going to design a Gibbs sampler algorithm. And here I said prototype because this is meant to be not an efficient algorithm. It's definitely not the algorithm that we use. It's purely for pedagogical value. So the idea is that, well, okay, at each step, pick a random organism in your system, reassign it to an existing cluster according to the Chinese restaurant process reweighting I told you about. And then you're going to, again, resample the alpha, which is the parameterization on the Dirichlet process, conditional on the new clustering that you have. And then you're going to pick a random GLV parameter and resample them based on all the other things in the system. And remember, these coefficients are meant to be either truncated normals or normals. And I just told you about how to do the first two, because we have a Dirichlet process and it's kind of nice. Turns out that 2C is also nice, but you do have to put in a little bit of work and think about what it is actually you're doing. And the reason it's different is that, well, let me go back here for a second. So you can resample the top part, but when you resample the clusters, you're resampling these parameters. What you're actually doing is, well, for the GLV system, you want to think about how likely it was that you saw this relationship, right? So the difference in the logs is approximately equal to this expression, which is the derivative in the GLV system that we wrote down, maybe plus some small Gaussian noise, right? So so it's going to turn out something like, if you write down the equations, if you and look at what it looks like for a Bayesian ridge or a lasso, it's going to look very similar. And we'll get there, but before that, there's some considerations to make when you're designing an MCMC algorithm, which is, you want to think about its efficiency, how long it takes. Now, this prototype algorithm that I just showed you is very slow mixing in the sense that, when I talk about Markov chains, mixing just means how long do you have to run the Markov chain in order to forget the initialization. Now, because you're doing one parameter at a time, you have to do it many, many times to even touch all the parameters to begin with. So that's at least n log n steps, according to the coupon corrected result. And the other thing is, well, when you're reassigning clusters, it sort of depends on the simulation or the prediction that the GLV system gives. It's not just one cluster in a vacuum and that you're reassigning, if you reassign a cluster, then it sort of affects everything else because it's a big dynamic system that you want to look at as a whole. Now, there are ways to address each of these problems, and the first two are kind of free because maybe the algorithm was not smart because we're not sampling all the variables at each iteration. Maybe you can do something like shuffle the order in which you're resampling the variables, but make sure you do all of them. And also, instead of fixing one and sampling all the other ones, maybe you can sample jointly. The third one is the most important that for us, which is you want to marginalize out parameters using their conditional distributions whenever possible. So you're not stuck sort of in a local regime where your reassignments are considering all the current GOV parameters, which actually might be very sensitive. And I'll explain what I mean by that. Well, let's skip that. So when we talk about this marginalization, what we actually mean is that think about the cluster reassignment step. And I swept this under the rug. So this model likelihood is actually kind of hard to compute. If you think about it, it's not hard in the sense that it's mathematically difficult. It's a little computationally burdensome. So think about what it is you're doing. Well, again, you have this system, the GLV derivative here, and the difference in logs. I just moved the previous time point to the right-hand side. Now, if you think about what you need to compute in order to perform this update step, well, in order to resample this new cluster, you think about the likelihood, this whole thing, since this noise is a Gaussian term, you're fixing this log x term, you're fixing this xi term, and you're fixing this xj term. And maybe because you're doing Gibbs sampling, you're also conditioning on the other coefficients like ri, ai. Now, the only thing you're trying to change is whether or not which cluster, the only thing you're trying to change is which cluster I is in, for example, at each decision-making process. Now, if you're trying to do this, well, you can either resample a new cluster with everything held fixed, or you can choose to say, OK, we're going to fix everything, but we're going to still think about these as being random, the coefficients ri, ai, and b's. But we're going to consider their conditional distributions based on everything else. So this latter viewpoint is going to mix faster because you're going to be sampling from the full conditional distribution and not just the current value that it's at in the Markov chain. And let me pause for a second for questions. I take a sip of water.
I have a question.
Yeah.
Which is, what controls the variance of your noise epsilon?
So in this toy model that I'm presenting, it might be helpful to think of it as fixed. A priori, you don't actually know what it is, so it might be helpful to actually put a prior on the variance. So that's actually what we have done. And turns out that the conjugate choice for the variance term for a Gaussian is, let's say, for example, like a random variable that's 1 over a gamma, or 1 over a gamma distribution. Or maybe there's explicit way to write down the distribution over the reciprocal of a gamma. So it's turns out it's a rescaled version of chi-squared distribution. But you make either of these choices and it might be a good prior for the model. Right. So the short answer was that here in this example, it's maybe you could hold it fixed. Okay. Now you can start to do more interesting things with this, and you'll see this in Travis's talk, which is we've sort of designed a simple model, gone one step. Maybe for the sake of interpretability, we want to explicitly model which interactions exist or don't exist. So one thing you can introduce is maybe a Boolean flag here, take its values 1 or 0. And maybe this is supposed to represent either a presence or non-presence of an edge in the interaction graph, for example, right? So if you just had this, you might be stuck interpreting, well, which values are small. If you see a posterior with only small values, you might be stuck interpreting, well, is this interaction real or not? And you can explicitly model, is it better for the model to have this interaction or not? And that's essentially all I've had to say so far. And the takeaways are that, well, when you're interpreting, when you're trying to learn even these really complicated dynamical systems, it might be helpful to use structural assumptions. And this is a classical technique in pretty much all of over-parameterized machine learning tools, right? Statistical learning tools, which is you want to regularize, you want to think of it in terms of regularization and exploit any sort of commonalities or low complexity substructures in your model. And for these questions involving like, why did you choose gammas, or why did you choose these certain priors, and why did you choose a Dirichlet process prior? It turns out that if you make certain choices, it admits faster algorithms, and it even increases interpretability if you do it properly. For MCMC algorithms, you can do lots of things like marginalization and joint sampling to try to speed them up a little bit. And you should use everything in your toolbox to try to do so, especially because you're trying to learn these extremely large dynamical systems. Remember, think microbiome, not hundreds, but maybe thousands of organisms. And that's it for the talk. Here are the references, and thank you very much.
Thanks, everyone. And thanks for coming in person for the crowd. It's fun to start to get back to normal. Also, to just sort of bookkeeping, I know there were some questions in chat about specifics of Lodka Volterra, so don't forget to bring that up. And I know that they were in chat, so maybe you can mention them over, like orally, to talk about those, because I don't want to forget to address those questions. Okay, right. So the motivation for today. Oh, yeah. So first of all, I have no conflicts. I would like some. The main sources of funding are from Georg Gerber and myself. And then you just had a contributed talk from Younhun Kim, a research associate at my lab, so also contributed heavily to this work. And then this is kind of bleed over work from when I was still a postdoc. So the other major contributor of this work who's not here is Georg Gerber, who was my former postdoc advisor. Right. So what's the motivation for this work? So the motivation is this example for bugs as drugs. And so here's a nice little example. Let's say we're studying C. diff infections. We know that you can be an otherwise healthy person. You take antibiotics for some unrelated issue from your gut. And because you've disrupted the normal microbiome, you might have a bloom of C. diff, and you can get a C. diff infection. And so this was kind of work that predates when I started working with Georg, but they were already starting to look at these model organism systems. So they would take 10 to 20 taxa, they would put them into germ-free mice, they would challenge the mice with C. diff, and then they would try to learn these small ecological networks, or as Georg likes to call them, pharmacology meets ecology, to try and figure out how they can make microbial therapies. So you might add then in this cocktail, not only the bug that suppresses C. diff, but maybe the bug that supports the bug that suppresses C. diff. So you could sort of have this robust bacterial therapy that you could take, as opposed to C. diff, which in some cases isn't very well characterized. And so they consider this to be like a safer option and more of a direct therapeutic for treating C. diff. And I will just say that, and Lynn Bree has gone on in her group to actually fully characterize this, and they think that they've actually uncovered the mechanism for developing bacterial therapies for bugs that you can add where there's 100% lethality when you have a C. diff infection in germ-free mice. And then they can also add another bug that is 100% protective against C. diff infection. And it all has to do with stickland fermentation. So if you're interested in that work, I suggest going to some of these papers below. I think they're both out in Cell Host and Microbiome recently published it. Okay, so that's kind of a simple scenario though. The mechanism of C. diff is sort of well characterized. We know that it's an infection caused by a single bug. But for things like IBD that aren't well characterized, we are starting with a much more complex system. There's also these host effects. We're not sure what component of the bugs contribute to these diseases, and it's also a plethora of diseases. And So it's overall not well characterized, but let's say we wanted to take the same approach. We wanted to start to build bacteriotherapies for things like IBD, but we were starting in a much more complex scenario. How could we sort of do this pharmacology meets ecology? And in the first talk, you kind of heard about the ways that we were thinking about scaling these dynamical systems to address this problem. And I will talk a little bit about the application of such models to try and head towards the direction of studying complex microbiomes and looking at their dynamics. Okay, so this is the general setup. We're going to be learning from time series data. And I just wanted to point out here that we, in our studies, we usually have, where'd my mouse go? Can you see my mouse? It has disappeared. Okay, on the left-hand side, oh, they won't be able to see that because they're looking at the screen. Okay, never mind. Okay, maybe I'll do it for the benefit of people here. Does this work? Oh, it does. Okay, so I'm pointing on the left-hand side here. Notice we have two data modalities. So we do this because, with sequencing, read depth is not quantitative. So you actually won't get the entire bacterial load from either amplicon sequencing or shotgun sequencing. So in addition to doing sequencing, we also do qPCR measurements as well to try and get a handle on the total bacterial load. Especially with the kind of perturbations we add, the bacterial load can change significantly, and we don't want to just be left looking at sort of relative abundances when we know that there's large changes in the entire population. Other groups have now started, and we have started to look at as well spike-ins. That's another way to get a quantitative measurement, because you can spike in a known quantity of a bug that is not in the community, and then just do regular sequencing, and then you can back out the total abundance. And then another thing just to notice that makes this challenging is not only do we have a mixture of different measurements with their own sort of uncertainty associated with them, the measurements are sparse and irregular because we're usually looking at fecal samples when we're looking at these mouse models. And even with humans, you can't generate infinitely dense time series of fecal samples. There are some physiological constraints on that. And then the idea, once again, is that we would like to go from these time series to these sort of interaction networks. And so very briefly, I'll go over then, for those of you who weren't in the first talk, what do we do when we try to model interaction networks? Well, first, let's just start with the simple system of one bug. And the easiest thing to do is logistic growth. So there's two terms here. The blue alpha is the growth rate, and the beta II is the sort of self-limiting term. And so you can see on this plot, there's exponential growth, and then a carrying capacity can be backed out of that. And then if you just want to make this equation one step more complex, you add these beta ij's, which are then interaction terms, which you can think of as edges in this graph, right? So any questions on that so far? Just sort of kind of the most direct way to get at this interaction network if you weren't here for the first talk. Okay, no questions. All right, please ask questions. Okay, yeah, but now what if there were hundreds of taxa? Let's see, Younhun said hundreds, maybe even thousands, but even if there's only 100 taxa, it's still a really hard problem. I mean, how many edges are in that graph, right? So if you have 300 bugs, it's like 90,000 edges of potential interactions between the bugs. And that's really what we want to address, is the problem of, if we just take a straightforward and naive approach, we don't want to be staring at this picture. So let's say we had the time series data, we did a cool experiment. We wanted to learn these beta ij parameters, sort of a straightforward like regression approach to those dynamics. If we discretize them, you would very, very quickly run into this sort of protein interaction network type figure. And so sort of before doing these experiments, our big contribution in this ICML paper was to come up with efficient ways to model microbial dynamics, to automatically learn the structure in terms of modules. And so once again, here, if the taxa are in these yellow, purple, and orange in the bottom right, we think about these modules as working in concert so that everything in this module interacts with everything in this other module in the same way. Right? And as kind of addressed in the previous talk, this makes inference more efficient. Just to give you like a heads up on the time scale of doing inference. So doing 15,000 Gibbs steps with this model for 100 taxa with 700 time points takes about a day if we do it this way, and if we do it the other way, well, we just stop the simulation because even initializing the model is kind of hard. Like once all the interactions start coming on, the first few Gibbs steps are extremely slow before it tries to start to sparsify the network. So it takes a week to a month to run it without having this module learning turned on. Okay, and I'm just going to give you a heads up view of everything that we're going to go over. So I'll talk about the types of gnotobiotic experiments we do and the time series that we have before that we feed into what we're calling mDesign 2 that learns these modules, which is a fully Bayesian model. And then what we do, instead of looking at the interaction network in terms of individual taxa, we look at it sort of at this module level. So this module interaction network. And then we can sort of do everything at the module level, which is a little bit easier to understand and interpret. Okay. All right, and so really quickly, just going to talk about some of the model components here in terms of the logical-Volterra dynamics and quickly build up and show you where we're headed before getting to the results is, so the first thing we do is instead of looking at the pairwise interactions between all I and j, as Yun showed you, we look at the interactions using these modules or these clusters of bugs, which you see by the CI/CJ. We're also going to have multiple perturbations. So we do this because if you just let the microbiome, well, I'll talk about specifically the mice because that's what I know pretty well. You know, after about a week or so, the dynamics pretty much stabilize, and without any perturbations, you're not really going to be learning any more dynamics, just characterizing the sort of noise of the system. So we do successive rounds of perturbations to not only make the measurements to have rich enough dynamics to even learn things, but it's also a more efficient way to use the mice because we can use these successive rounds of perturbation as opposed to having multiple cohorts of mice. So we get like, 3 or 4 experiments into one essentially, in terms of how many perturbations we can do over, a 60 to 80 day window. And then the last part that we add, and there's more to the model, but we also add sort of these structural learning components where we explicitly add the indicator functions to the variables of interest, like the perturbations or the module interaction terms, so that we can compute things like Bayes factors and things like that. So we can know how important certain variables are into being able to, how important certain variables are in minimizing likelihood.
So quick question, Travis, are
Yeah.
the perturbations always at the level of individual taxa or groups of taxa, or what's sort of the level, what's the level at which perturbations are done?
There's a decision to be made there. We thought about it. You could make it so that the perturbations did not respect the other modules. They could have their own sort of modules that were distinct from the other modules, or you could not group them at all. We decided to group the bugs by perturbations as well. So not only do the modules interact in the same way between each other, but if you go on a high-fat diet and the effect on that module is positive, then everyone in that module has a positive effect from the high-fat diet. Yeah, and so there's lots of different ways you can think about changing this. You could of course let them be in their own modules. You could not add the module component. You could even change like the notion of what a module is for us. No one's asked it, but a good question is, you've set it up so that the modules work in cohort where no one, explicitly the way it is, if you're in a module, you don't have any interactions with whom is in the module that you're in, right? Because if you were interacting with them, they would be in another module. So you could sort of flip this around and say, no, why don't we just make everyone who's in a module only those bugs that are interacting? And so that's actually something that we're trying to address with another project where we're going to have, it's not spatial sequencing, but we embed the feces into small particles, and then we sequence the particles in bulk. But the idea is that we know that how big that particle is. So it's kind of like spatial sequencing, and we can then look at microbial abundances sort of spatially as well to try and test out, who's close to who and who's interacting with who, and we can potentially sort of flip these ideas on their head. But yeah,
are
really
the experimental perturbations always on a more global level, like
it's just
diet
to the mouse. Yeah. So the perturbation is the diet or antibiotics. So potentially, it's whatever's going on in the mouse. Yeah, we don't target the perturbations to specific taxa. So yeah. Right, and then just sort of like a quick plug on why we use these Bayesian models.
Sorry, quick question. On the previous slide, these modules are defined entirely from phenotype?
Yeah, from the data. Yeah, they're
okay,
inferred.
So
from the
there's no sort of prior based on relatedness.
Yes, another great question. Something we also are going to add at some point. We didn't see strong phylogenetic similarity between the modules, but because it is a Bayesian model, you could definitely put a prior based upon phylogeny, right? You know, that's also something we've been thinking about. Another great question. But because we're only looking at the 16S gene, usually when we build these phylogenies, you know, is that really characteristic of the total functional capacity of the bug? So that's actually something we want to test, or if anyone is interested in testing that, you know, does having a phylogenetic prior actually make the model have better predictive capability? That would be cool to actually test, but we did not add that here. But yeah, another good question. Yeah. So yeah, and then just a quick plug. Why are we doing these Bayesian models? Well, because of this scenarios 1 and 2, right? We don't, you know, lots of techniques that are fast. And sort of the first thing you would do is you do sort of map estimation or point estimation, like if you're doing a regression problem. But we really, we know that we have a really noisy system and we're mixing multiple data modalities. So what we would like to do is to propagate that uncertainty throughout the model so that we can get an idea of, you know, imagine if this was a growth rate in purple. In one scenario, you don't know, essentially the growth rate could be really small, could actually be negative in the model if you allowed it to be. Whereas on the right, it's very clearly, you know, a positive value. And we don't just do it by looking at these distributions. Once again, we add these indicator variables that can kind of help actually explicitly say when something is important to the model. But it's just sort of a plug of why we're using these Bayesian models, because we're mixing, you know, multiple different levels of uncertainty from the measurements. And because the data is so hard to get, we might as well spend the time to make these fully probabilistic models. Okay, and then here's basically the nuts and bolts of the basic parts of the model. I've already kind of shown you what the dynamics are like. Sorry, I dropped. Yeah, here it is. So I'm looking at the dynamics box for those not in the room. We've kind of already introduced this. Young gave a version of this. These are just the priors on the growth, self-limiting, and the perturbation effects. These are, you know, normal distributions. The priors for the indicators are Bernoulli. Jan went into great detail about the prior we use for the module assignments, and so that's a Dirichlet process. And he's, you know, we did that for 2 reasons. One is we have an efficient way of actually sampling what alpha is. So there's a hyper prior for alpha, and then alpha indirectly sort of determines the number of clusters. And because we don't want to a priori specify the number of modules, that's important to us. And then one thing that Jan didn't talk about at all about, but we actually have a measurement model as well. And I'm actually going to go into a little bit more detail about that in the next slide. So having a little bit of history, when these modules were first used, they were used within 2 contexts. So one was Vani Bucci's work and Stein, and what they initially did was they were looking at sort of the time series from, you know, Eric Alm's group had this time series over about a year, I think, and I think Rob Knight had one as well. And so they were just looking at those time series in a human and trying to learn microbial dynamics. But what they quickly realized is that they could really only, like when they would get a network or when they would try to forecast the dynamics, they could really only look at the top 10 or so bugs to reliably see what's going on. And I just wanted to point out that, you know, if you own, like the top 10 bugs take up 99% of the reads. And so actually, and I'll try and show this later, that, you know, a lot of the interesting stuff is going to be going on, you know, this is just the top 100 bugs, but, you know, from bug at the 90th percent, you know, down to maybe 10% or 5% of the reads, you know, those are the bugs, you know, that's the abundance that, you know, E. coli is at, and that C. diff is at. And all of these other bugs that we might even be interested in are all in this sort of mid to low abundance regime within the top 100 bugs. And they have lower, a smaller fraction of reads that are apportioned to them. And so that's kind of why we think it's really important to have a noise model, because the kind of bugs that you care about inherently have more noise in these types of models. What did we do for the reads? Well, we just have this negative binomial distribution, which is just a Poisson with a prior of the gamma prior, and then you integrate it out. And so it's this sort of over dispersed model. You can kind of see the over dispersion here at the bottom. Oh, is my mouse working now? Oh, yay. And then for qPCR, these are just log normally distributed. They come in triplicates. You generate a standard curve, use the plates. And then the last thing I'll say is that because these are already come in triplicate, you can get a measure of the variance. Here, what we did is we actually took, we don't have replicates for every single one of the time fecal samples for the reads, but what we do is we take a fecal sample, or we take an individual time point, and we take the mouse. And if we have 2 or 3 fecal samples at that time point, we then take those physical pieces and we make them even smaller, maybe into triplicate as well. And if there's 3 pieces, we make them each into 3. We have 9 pieces, and we run all 9 through our pipeline. So from DNA extraction onto sequencing, and then we use those 9 to fit the parameters for the negative binomial distribution. So those parameters for the negative binomial in the qPCR are held fixed. The variance in those model parameters are not updated during inference, whereas all the other parameters in the model are. Okay, and then just for the uninitiated, we had a Dirichlet process prior on the module assignments. And so one interpretation of it is that the Dirichlet process is what's known as this Chinese restaurant process. In the absence of any other data, like the likelihood for the dynamics, what does this model do? Well, it's just sort of a rich get richer or popularity contest. If you're the red block and you show up to a restaurant and you're looking at tables, the probability of you sitting at a table is proportional to whoever's there. And the probability of going to a new table is proportional to alpha, which is just the concentration parameter. So the concentration parameter then explicitly models how likely you are to jump to a new table. Implicitly controls the number of tables. And since we don't a priori know how many modules there's going to be, alpha is not fixed, but alpha has a prior, a diffuse prior. Okay, and so this is the complete model. Some people like looking at these. I've never been able to look at these and understand exactly what's going on, but some people can. So that's for you, just to show that this is a fully Bayesian model. Noise propagates through the entire thing. And you see our measurements are down here. These are the, I lost my mouse again. These are the Y's and the Q's, our reads and our qPCR measurements. Okay, right. And then once again, this is we used MCMC on this. And so once again, Younhun kind of already went over this, but the way that we're doing sampling here is, let's say when we're looking at the edges, we turn an edge on and off in the graph. We look at the likelihood ratio, and then based upon how likely that is in the model, it chooses during that sample to be stochastically to be on or off. Same thing with the cluster assignments. You run each taxa through all the modules. You calculate how likely it is for you to be in those modules. Once again, you sample from distribution, and you might move into a new module or create your own, and so on and so forth. And then here's just an example for 1,000 Gibbs steps. So this is the growth rate. There was a question about how well the system mixes. So this is the growth rate. It's a constrained to be a positive, so it's a constrained normal distribution. We do calculate statistics like the R-hat statistic from different runs, but here you can see it's, just by eye, it's a fairly well characterized normal distribution. And variables like that mix pretty quickly. They're the easy ones to learn. Okay, so now I get to go to the fun stuff. All right, so let's start to talk about the results. So here's the setup for the kind of data we're getting. So we had 2 cohorts. We had a healthy cohort of 4 mice, and the dysbiotic cohort. It was from a UC patient. So we had 4 mice in the healthy and 5 in the UC cohort, and each cohort had sort of 1 donor. So even though we have 4 mice and 5 mice, we had 1 donor in each group. So if you're reviewing our paper, just forget that. Small facts. I think at the time it was like $100,000 experiment. So if we wanted to scale this to even more people, at the time it wasn't really feasible, but we're definitely following up on trying to scale this to even a larger set of donor samples. And then we have 3 successive rounds of perturbation. We have a high-fat diet, vancomycin, gentamicin, targeting different groups of taxa. And then the one point I'll state is that you always have a budget, and so we were actually careful about the density of sampling. So we took more measurements or made measurements closer in time just before perturbation and immediately after, because that's when you expect the most transients. And then after 3 or 4 days, then we went to every other day sampling. So twice a day sampling just before the perturbation and for the 3 days after, and then every day. And then if it, we tried to set it up so the weekends would be pushed out at least, 5 days from the perturbation.
Travis, quick question. Why is a high-fat diet of interest as a perturbation?
Just because from experience, we know it has sort of a massive change in the abundances of the bugs that it targets. So there's been studies even in this area where they've looked at things like, high protein diet, high fat diets. And just, we know that the high fat diet seemed to have the biggest change amongst of the broadest set of taxa. And so that's kind of like, what's the best hammer you could use? And then we also staggered it too. You'll notice we did the high fat diet first because we expected it to sort of remodel the cause the biggest transients. And then we did the antibiotics because we didn't want to lose any taxa originally. So we kind of sort of carefully did it that way. Turns out gentamicin wasn't the best antibiotic to use. It doesn't really cause that broad of a change, but the vancomycin definitely causes a pretty big change in the bacterial load of the bugs.
Travis, just a quick question here. So you have like 2 cohorts, right? Like the healthy and the UC sort of mice, and then you introduce perturbations on them, right? Like either. And what is the, like, are you mixing them or like, that's what you're going to show here, I guess, right? Like
Right.
Okay.
Yeah. So the experimental design was the same in terms of the perturbations. There's no mixing of the cohorts. The cohorts were to be separate.
Sure. I meant the mixing of perturbations, like high fat, 2 different antibiotics.
Oh no, they were done serially one after another, right? So here you can see on day 21 or 22 is a high fat diet for 10 days, 2 weeks off or 10 days off, vancomycin, and then gentamicin.
Okay, got it. Thanks.
Yeah, and then one question could be, does the ordering matter? We don't think so. We'll probably have to test it at some point if we keep on doing this kind of work. But also adhere to be efficient. If we wanted to do each of these independently, you would have extra cohorts of mice for no other reason than to just give them a different perturbation. But we're trying to show here that, we think it's safe to do these perturbations serially.
Got it. These perturbations are matched for both the healthy and UC cohorts, right? Like you do the exact same thing.
Yeah. 9. Yeah. So So all the gnotobiotic, we actually start the experiment at the same time. These might have even been from the same litter of mice, but then they go into their isolators or optimized cages here because it's complex flora. But yeah, the experiment is run at the same time, and the perturbations are at the same time and last the same time, and the fecal samples are collected at the same time.
Cool, thanks.
Yep, awesome. Thanks.
Hi, Travis. I just have a quick question. So do people know how representative the fecal samples that you collect are of what's actually going on in the gut? Or are there like some bugs that are fixed somehow in the gut and aren't actually coming out?
Oh, you mean, yeah. So have there been studies where people have taken sections of the gut and looked at the bugs? So what's in the bug what's in the feces I know is most representative, what's nearest the end of the gut. But I'm not an expert at all on the sort of physiology and the biology of which bugs prefer which niches. No, that's definitely out of my wheelhouse. So if anyone else knows the answer to that, chime in. Okay. Yeah, and then so one of the things we did is, we've seen in these studies with healthy patients or patients over time, both healthy and dysbiotic, that there's this sort of difference in sort of diversity. And here we're just looking at alpha diversity. And so we were happy to see it was recapitulated in the mice because the first question you could say is, okay, great, you took this UC in healthy fecal samples. Do they have any of the characteristics that you would expect that we know about humans when we just look at the stool from the human directly? And so it's well represented here that, across the board, the healthy microbiome has higher alpha diversity, During the perturbations, you actually get this nice thing where there's like much more uncertainty. So actually, there's some dynamics and there's some transients going on, which is why you see the larger error bars. But then after each successive round of perturbations, it returns to this sort of, I don't know, steady state alpha diversity. It returns to the type of diversity you would expect based upon the different cohorts. Right, and then before getting to the result, this is the last part about the model. I wanted to bring this up because, when we tried to show how good our model was compared to other systems that are just doing point-wise estimation for the parameters and doing forecasting, we got these results on the left. So what you're looking at, the results on the left is how well our model predicted relative to 2 other models, where we train on 4 of the mice and leave out 1 entire time series. We start with the initial condition on day 0, and we simulate for 70 days. And then this is the error in terms of the log of the root mean squared error, or the root mean squared error of the logs. So we were like, great, statistically significant, but if you just look at that figure, you'd be like, well, was it worth it? But when you start to break it down in terms of the abundances of the bugs, you can really see where our algorithm excels. And so at the far right, you can see this is 10 to the 9 to 10 to the 2 about. Our algorithm isn't statistically significantly doing better at predicting those. But then when you look at, from 10 to the 9 all the way down to 10 to the 6, that's where the extra oomph in terms of the accuracy of our algorithm is on predicting the abundance of the bugs in the held out data. And so that's sort of the advantage of the noise model. And then, it actually doesn't, you're not even able to do a sort of a stochastic GLV model with MCMC if you don't do the modules. So the noise we think helps, and the modules are there to make it an efficient process. And we're still able to forecast better on the larger fraction of the bugs that aren't in the top 10% that take up 99% of the reads. So I thought that was cool, and why it's important. I've learned this, to look at the larger fraction of the lower abundance bugs and to actually break it out by these quantiles or groupings, because otherwise, if you just group them all together, you kind of miss this completely. Okay, and so this is the beginning of the kind of pictures you can start to get. There's not a lot to be taken away from this, just to say that you can construct a phylogenetic tree of which groups of bugs are with which group of bugs, and you can start to get an idea of this the module interaction network. Once again, we don't interpret this directly. This is just sort of a snapshot of what it gives you. It tells you how all the cohorts are responding to the perturbations. And things like that. One interesting thing to note we saw is that the high-fat diet, it's kind of hard to see. You'd have to look in the paper, but the high-fat diet only had a positive effect, which is nice. So if you think of a direct effect, it's like changing nutrients. High-fat diet isn't going to necessarily kill bugs. But then all of the antibiotics, the ones that had high base factors, were all negative results. So it's nice to see that the intuition is that you would expect an antibiotic to be a direct negative effect on the growth rate, and maybe like a dietary perturbation to selectively allow for other bugs to grow. That was nice. And then I'm going to go into more detail on this network, so don't pay attention to this network yet. This is just kind of getting our feet wet on the results. Also, and this was kind of an interesting result we didn't expect. So between these 2 cohorts of mice, we had about 60% overlap in the OTUs. So that means that, about half of the OTUs that were in each cohort were not representing the other one. But we wanted to address and see, is module 1 in the healthy cohort, does it have a module in the dysbiotic cohort that is very similar, maybe phylogenetically? And so what we did is we did sort of this agglomerative clustering where we started successively grouping bugs together that were phylogenetically, we started collapsing bugs together that were had a higher probability of clustering together, and then we looked at how correlated those clusterings were across the cohorts, because eventually you get to a point where there's one sort of mega representative module, and so it's a way of kind of finding common agglomerations of the different modules. And just to say that it looks like these modules are sort of there's like a thumbprint to the modules. If it has a certain representative taxa, it's not going to be the same one in the other module, but it's going to be phylogenetically similar. And so these modules do have sort of these signatures where they look similar. Yeah, I haven't figured out the best way to describe this yet. And then what's interesting is that this sort of, it's statistically significant up until about 85% identity, but then that's about the family level, right? And so what you see is that there is sort of this phylogenetic signature to each of the modules, even though the modules themselves aren't phylogenetically coherent, right? They're like not all from one specific genus or going up, but they're sort of represented in the same ways up until you get to the family level. And then once you get to the family level here, sort of all the phylogenetic coherence is gone. So yeah, that's kind of interesting. I know I didn't explain that very well. This is like the one part that I'm not totally comfortable explaining because it's kind of complicated what we did. But let's get to the results. Okay, so now we have these networks and we can start to do fun things, like we can start to perturb the dynamics and then simulate forward. And so Younhun started to address this, which was if you perturb a fraction of the OTUs and then you simulate forward after you've perturbed them, do you return to the steady state? And so you can already see that for the blue, which is the healthy, when you perturb the same fraction of OTUs, yes, it doesn't always return exactly to the same steady state, but it returns closer. So the orange is further away and the orange has a larger spread. So not only in the dysbiotic setting, do you stay further away from your steady state, but you have higher variability in where you go. You can look at it in terms of either steady state or entropy. Lower entropy here is what you would expect for the UC or the dysbiotic state. And once again, it's the same thing as recapitulated. You have lower entropy, but higher variability as well. Maybe suggesting something like instability. And so then we went along and we tested that. So, so there's actually, I guess the big takeaway here is we actually looked at the networks and we said, do you have sort of these features that we would expect from an unstable system if we looked at sort of properties of feedback cycles? And so the only sort of feedback cycles that are stable for all sorts of interaction strengths are these negative feedback cycles. So it'd be like a plus minus relationship that you see at the top. And in all other scenarios for 2 cycles where there's positive feedback or higher order cycles, you expect there to be the higher, like there is such a feedback strength such that the system can be unstable. And then what we saw on the dynamics was precisely that the only time the healthy dynamics, they had more of these negative feedback cycles, sort of this plus minus, but across the board, we saw more competition and higher order cycles in the dysbiotic state. So this notion of like what we saw in the forward simulations can be sort of analyzed from this dynamical systems perspective on the slide here, and those two are consistent. And if you also look at sort of the eigenvalues of the interaction matrix, it's also consistent. I don't want to say too much more in here because there's entire groups that study this stuff and ecologists that study this in detail, just to say that this is all consistent with the previous slide. And then sort of the final result and how we actually want to use this is, you know, what did I say in the beginning? I said, we want to sort of be able to do what we did in the simple setting where we had 10 or 15 bugs. And we wanted to try and understand who's interacting with who to try and build, you know, bacterial therapies. And so looking at the module level, what we then did here is we did the keystone analysis where we removed modules one at a time, simulated the dynamics and saw who interacted with who most strongly in terms of like the biggest disruptions to the total population. And so then we found some interesting things. And so in the healthy state on the left, we found that the sort of most important module in terms of maintaining the abundance of all of its neighbors was this H9 module. It has a group of bugs, all are starch degraders, and in particular, Ruminococcus bromai and Gemiger are RS2, RS3 resistant starch degraders. And then they perform this cross-feeding chain. Well, okay, we haven't proved it yet, but it implies some type of cross-feeding chain to these gram-positive bugs, which then cross-feed into the gram-negative bugs, or at least that's what they're suggesting. And then on the right, you have a totally different picture where you have lots of competition between bugs. Ruminococcus bromai and Gemiger are not present. They're actually, and if you look at the HMP2 cohort, Ruminococcus bromai is overrepresented, I think, in the dysbiotic cohort in that study as well. And actually, there's been some literature on Ruminococcus bromai already being called a keystone taxa. But on the right, you have a very different story where you have lots of competition between these like Lachnospiraceae and Ruminococcaceae. And also a different story in terms of what's the most beneficial module in the dysbiotic cohort. In the dysbiotic cohort, there isn't some module sort of acting like a cross-feeding source for everyone. It's actually sort of the enemy of my enemy is my friend. It's actually the removal of someone in competition who's competing with everyone else that actually allows for the biggest growth. And another module, which is interesting, is Pseudorella-Wadsworthensens, you see in D10. It degrades host IgA. So that's also been thought of as a pathobiont. Because it degrades host IgA, it allows other pathogenic strains to hang out and your body is less likely to fight them off. And so, yeah, so it's interesting that these findings were kind of consistent with what had already been in the literature. Now, I guess the main thing that we're doing is we now have, modules which we're targeting to actually do follow-up experiments. So kind of like what we did in those previous studies where we have smaller communities of bugs, we now have sort of a two-pronged attack where we have groups of bugs we want to add to dysbiotic microbiomes to see if we can sort of adjust the diversity profile, as well as simultaneously making these more defined communities that were done in the earlier studies, but by also adding in these, like starch degraders and maybe adding in Pseudorella-Wadsworthensens. But not only those bugs, but the community of bugs around them that'll make for these sort of like stable or robust, either healthy states or dysbiotic states. Yeah, and so that's all I really wanted to show with you, sort of our sort of two-pronged approach. We came up with this new model that actually allows us to do this at scale with a fully Bayesian model that we couldn't do otherwise. Like we couldn't propagate the noise unless we learned these modules. We also introduced these sort of humanized mouse models where we could start to study how the bacteria are interacting with each other in these mice. Yeah, but still lots of work to do. So, we haven't validated these interactions, but we're using this model to help prioritize where we want to spend those resources next. And we also found interesting characteristics in terms of stability between the two, which are consistent with what people have seen or seen with some evidence in other studies looking at the human microbiome as well. All right, and with that, I guess I'll take questions.
Yeah, so thank you so much, Travis. Let's thank the speaker.